as we move into the A2 content, you will find more and more areas of overlap with topics that you've already covered in the earlier modules. So, for example, to understand hormonal communication, to understand the nervous system, it's really important to remember how things move across a semi-permeable membrane. Diffusion is the movement of molecules from where you have a lot of them, a high concentration, to where you have less of them, a low concentration, across a semi-permeable membrane. And remember that a cell surface membrane is made up of a phospholipid bilayer. So you have two layers of phospholipids with the hydrophilic heads on the outside, which are phosphate heads, and the hydrophobic fatty acid tails on the inside. Molecules are able to diffuse directly through the phospholipid bilayer if they are small and they are completely non-polar, so they don't have any charge at all. So, for example, oxygen and carbon dioxide can freely diffuse across the phospholipid bilayer. Be careful with the terminology you use in an exam. They do not diffuse through the phospholipids. That implies that they break apart the phospholipids going through the membrane. What you say is they can diffuse across the phospholipid bilayer or diffuse through the phospholipid bilayer. Water is not non-polar. Water is very, very slightly polar, but because it's only slightly electronegative, it's able to diffuse slowly through the phospholipid bilayer. Sometimes you need water to be able to diffuse across faster, and at those times it makes use of specific transporter proteins called aquaporins, which allow water only to diffuse across them quickly. That's going to be particularly important when we look at the kidney topic. Molecules which are large or charged are not able to diffuse directly across the phospholipid bilayer, so they need to have a route through which they can travel, and that route is often provided by channel proteins. So they need help in order to diffuse through, and that help is provided via facilitated diffusion. If something is facilitated, it's aided. So in this case, the channel proteins are helping the molecules to diffuse across, but they're still going down there. In this diagram, they're still going down their concentration gradient. They're still going from a high to a low concentration, which means this process, in this example, is still passive. For facilitated diffusion to occur, the channel proteins have to be specific to the type of molecule that is going across the membrane. So channel proteins which are useful for allowing glucose to be transported will only allow glucose to be transported. They won't allow anything else. So these channel proteins are specific. You have different kinds of channel proteins which will allow different kinds of molecules to pass through the bilayer. This diagram is showing an example of active transport. And it's active because the molecules are going against their concentration gradient. On this side, if you look at these orange cubes as an example, on this side there's a low concentration. On this side there's a high concentration. These are the cubes I'm looking at. But the molecules are still being transported against their concentration gradient, which means this is active transport. It has to make use of energy in the form of ATP molecules in order to allow the molecules to cross in this way. Co-transporter pumps allow more than one thing to go across, but they will either carry two things across simultaneously, i.e. if one thing's going, there has to be something else there as well, or they'll transport two different things in different directions. A sodium-potassium pump is a really good example of this, and it is going to keep coming up. It's going to come up in the topic that we're doing at the moment for hormonal communication. It will come up a lot when we look at the nervous system. So what a sodium-potassium pump does is it allows 3 sodium ions to move out of a cell across the semi-permeable membrane, And once it's done that, it changes its shape, which exposes binding sites to 2 potassium ions, which will then move into the cell. Now would be a really good time just to pause the video a second and have a look at the information, the detail about how exactly this works. We're going to focus on the key important facts to remember about a sodium-potassium pump, which is that, first of all, this is an active process. It uses ATP. And for each cycle of the sodium-potassium pump, it pumps 3 sodium ions out for every 2 potassium ions that move in. 3 sodium ions out, 2 potassium ions in, and you do need to know which way round it is. Here's a useful tip to help you remember. 3 sodium ions out has 1, 2, 3, 1, 2, 3 characters in it, and 2 potassium ions in has 1, 2, 1, 2, 2 characters in it. Now imagine where this scenario is actually happening in a cell. You are losing 3 positive charges out of a cell for every 2 positive charges that are coming in. So overall, the cell is losing more positive charges than it is gaining. So overall, you're going to end up with a potential difference between the inside and the outside of the cell. Now, normally, to prevent a buildup of potassium ions on the inside of this cell, there would normally be some kind of channel proteins in the cell surface membrane where, as the potassium ions are pulled in by the sodium-potassium pump, some of them will then diffuse down their concentration gradient to move out of the cell. So they will be diffusing down their own concentration gradient through facilitated diffusion, but then they're actively being pumped back in by the sodium-potassium pump anyway. And eventually you end up with a balance of charge across the inside and the outside of the cell surface membrane, where it ends up being about minus 70 millivolts wherever this happens. Here's a simplified version of the diagram that's in your task sheet. And if this is a beta cell, then this is a cell which is in the pancreas, and it is a cell that should be secreting insulin. This particular task is about recognizing the level of detail about the mechanism of how an increased level of glucose causes insulin to be released. First bit is to recognize that you have a sodium-potassium pump, which is pumping 3 sodium ions out for every 2 potassium ions in. 3 sodium ions out, 2 potassium ions in, and that happens all the time. Now, it maintains a potential difference of about minus 70 millivolts because you also have these potassium ion channels which will allow some of those potassium ions to undergo facilitated diffusion and leave the cell down their concentration gradient. So you don't get a buildup of positive ions on the inside of the cell. It maintains some kind of an equilibrium. This is the version of the diagram that I gave you, but it's basically the same thing that I've just drawn. I've just deleted the bits that you don't need to look at straight away. So when you have a diagram like this, it helps to start with one point. In this case, you've got your sodium-potassium pump here, and you've got normal potassium ion channels, which will allow potassium ions to diffuse out of the cell. What you have to remember is that these potassium ion channels are ATP mediated, which means if there is too much ATP in the cell, these channels will close. As the blood glucose level rises, you have more than normal coming into the cell through normal facilitated diffusion through this transporter protein called GLUT2. And that means that that glucose gets used up in respiration to make ATP, which is what glucose is used for in a cell anyway. When that happens, These potassium channels, which were allowing potassium ions to move out, are now closed. And that means that the potassium ions can't move out anymore. That is now going to cause a buildup of these positive ions inside the cell. So whatever the resting potential was of this cell, it's not going to stay there anymore. The charge across the membranes is going to change because it was previously negative and now it's becoming more positive. It's losing that charge. It's called depolarization. The calcium ion channels, which are also within the cell surface membrane, are voltage dependent, which means that when the charge becomes more positive than it was, these channels are now going to open, and that will allow increased calcium ions inside the cell. Previously, there was a high concentration of calcium ions outside, but they couldn't diffuse inside because these channels were closed. Once the voltage is the right level, whatever that level is, these channels will open, they've had their signal, and that means the calcium ions can move in. The fact that those calcium ions are now moving in causes the vesicles which were holding the insulin to move to the surface of the cell membrane, and that causes exocytosis of the insulin, i.e. the vesicle fuses with the cell surface membrane and releases the insulin which was inside it outside. I strongly recommend watching that clip back and playing it forward step by step so that you can follow that chain reaction that starts with the blood glucose level rising and ends with insulin actually being secreted. Remember that if the blood glucose level is not that high, then you're not going to get that much ATP being produced in the first place. And if there's less ATP, the potassium ion channels will stay open. And that means that if they're open, you don't get a buildup of potassium ions. Therefore, the calcium ion channels won't open. Therefore, calcium ions don't move in. And therefore, you don't get exocytosis of the insulin vesicles.